Animals forage for food in ways that maximize intake and minimize their consumption efforts and predation risks. Foraging is the term used for the process of animals seeking their food. Animals typically have a wide range of foods that they exploit over the course of their lifetime. But in some cases, the food source that a species exploits is extremely limited. For example, giant pandas subsist almost entirely on the shoots and leaves of bamboo. Pandas must make decisions about which bamboo plants to harvest, which parts of a particular plant to eat and which to ignore. When Marla Sokolowski was working as an undergraduate research assistant, she noticed that some of the fruit fly larvae she was studying tended to move after feeding at a particular location. She called these larvae rovers, but others tended to stay put, so she called these larvae sitters. She let the larvae grow to adulthood, bred them, and found that the offspring of rovers also tended to be rovers, while offspring of sitters tended to be sitters. This suggested to her that the behavior is inherited. In fruit flies, variation in larval foraging behavior is at least partly due to genetic variation among individuals. Using information from the Drosophila melanogaster genome sequence and genetic mapping techniques, Sokolowski found and cloned a gene associated with a rover-sitter foraging difference. She named it the FOR gene, in reference to foraging. Follow-up work showed that the protein product of the FOR gene was involved in a signal transduction cascade, meaning it is involved in the response to a cell-cell signal. Rovers and sitters behave differently when they are foraging because they have different alleles of the FOR gene. Genes that are homologous to FOR have now been found in honeybees and the roundworm C. elegans. Researchers are investigating which signaling pathway uses the FOR gene product and how different alleles change feeding behavior. Presumably, the signaling pathway is associated with feeding or nervous system development. When this aspect of fly feeding is thoroughly understood at the proximate level, biologists will know why variants of the same protein make larvae more likely to move or stay in place after feeding. By altering population density experimentally and documenting the reproductive success of rovers and sitters, Sokolowski has shown that the rover allele in fruit fly larvae is favored when population density is high and food is in short supply. In contrast, the sitter allele reaches high frequency and low density populations where food is abundant. Rovers do better at high density because they are more likely to find unused patches of food that way. Sitters do better at low density because they don't waste energy moving around. These results are exciting because they link a proximate mechanism with an ultimate outcome. In this case, the presence of certain alleles is responsible for a difference in fitness in specific types of habitats. In Drosophila larvae, foraging is a relatively inflexible and innate behavior. Individuals don't decide to be rovers or sitters. It is the variability of food supplies that maintains both alleles in the population. But in Drosophila adults and in most animal species, foraging behavior is much more flexible and dependent upon learning. When biologists set out to study why animals forage in a particular way, they usually start by assuming that individuals make decisions that maximize the amount of usable energy they take in, given the costs of finding and ingesting their food, and the risk of being eaten while they're at it. This hypothesis that animals maximize their feeding efficiency is called optimal foraging. The specific costs and benefits of foraging depend on the feeding strategy and the environmental conditions of the organism. Different animals have different types of choices and thus different variables subject to optimization. For example, Antarctic fur seals forage by diving for small shrimp-like krill. They optimize their foraging by adjusting the duration and location of each dive depending on the depth and density of patches of krill. Shorebirds, called oystercatchers, feed on a variety of prey, including clams, which they have to open to eat. They optimize their foraging by selecting clams of intermediate size. Small clams provide less energy than large clams, but opening large clams takes more effort and it can damage the bird's bills.